What is the T1114 translocation in mantle cell lymphoma? Imagine a tiny swap happening deep inside your genetic code that can change how your cells behave. In mantle cell lymphoma, a specific genetic change called the T1114 translocation plays a major role. Mantle cell lymphoma is a type of non-Hodgkin lymphoma, which is a cancer that affects B lymphocytes, the cells that help fight infections. This lymphoma mainly targets B cells in the mantle zone of lymph nodes, but can also spread to the blood, bone marrow, spleen, and other organs. The T1114 translocation is a precise genetic event where parts of chromosomes 11 and 14 exchange places. Chromosome 11 carries a gene called CCN1, which produces a protein called cyclin D1. Chromosome 14 contains the immunoglobulin heavy chain gene, important for making antibodies. In this translocation, a piece of chromosome 11 that includes the CCN1 gene breaks off and attaches near the immunoglobulin gene on chromosome 14. This swap is written as T11, 14, Q13, Q32, indicating the exact locations involved. This genetic change causes the CCN1 gene to come under the control of the immunoglobulin gene's regulatory elements. Since the immunoglobulin gene is highly active in B cells, it drives overproduction of cyclin D1. Cyclin D1 is a protein that helps control the cell cycle, which is the process cells go through to grow and divide. When cyclin D1 is overproduced, B cells can start dividing uncontrollably, leading to the development of mantle cell lymphoma. The T1114 translocation is found in more than 95% of mantle cell lymphoma cases, making it a hallmark feature of the disease. Doctors often test for cyclin D1 protein expression using a technique called immunohistochemistry, which helps confirm the diagnosis. Sometimes, the translocation might be present even if cyclin D1 isn't detected with standard tests, so additional genetic tests like fluorescence in situ hybridization are used for accuracy. While this translocation is mainly associated with mantle cell lymphoma, it can also appear in other B-cell cancers like some cases of multiple myeloma and B-cell prolymphocytic leukemia. In the context of non-Hodgkin lymphoma, especially when the lymphoma cells are CD5 positive and CD23 negative, the presence of T11, 14, strongly suggests mantle cell lymphoma. Understanding this genetic change is not just important for diagnosis but also for ongoing research. Scientists are exploring treatments that target the overactive cyclin D1 pathway, aiming to develop drugs that can block this abnormal growth signal. For patients, knowing about the T11, 14 translocation helps explain their diagnosis and guides personalized treatment plans. In summary, the T11, 14 translocation is a key genetic feature of mantle cell lymphoma. It causes an overproduction of cyclin D1, which leads to uncontrolled B-cell growth. Detecting this translocation helps doctors confirm the diagnosis and differentiate mantle cell lymphoma from other lymphomas. As research advances, new therapies targeting this genetic change may improve outcomes, but current management relies on accurate testing and tailored treatment strategies. Always consult healthcare professionals for personalized advice and care.